Dear colleagues and friends, I'm addressing you from the Art Science Salon of the Mediterranean Institute for Life Sciences in Split, Croatia, that was this year proclaimed by Global Health and Pharmaceutics in England as the best European institute in the class of private non-profit institutes. I'm sad not to be with you in my beloved Paris and not to have occasion to talk with all of you. But I'm also sad for the reasons that I cannot be with you this evening. I wish only to express a point of view on biology of aging and of all age-related diseases. Thus, I want to improvise. No slides will be shown, and from time to time maybe a little bit of pantomime may be used. A provocative title could have been, "Why is biomedical research unproductive and boring?" It is provocative. But when we compare with the number of real discoveries of the major transitions in science and knowledge of the second half of 19th century and the first half, or the beginning of 20th century, with modern times, say with last half a century, if we normalized these discoveries or major transitions then with today, normalize them by the number of scientists involved and by the amount of money spent, then I think that today we are probably by 2, maybe 3 orders of magnitude less efficient in producing great discoveries, surprises, compared to what we have today. 60 years ago, maybe, the admired genius physicist Richard Feynman talked about observation, hypothesis, the principles and theories, and mentioned this. He said to his Cornell students, if upon observation you found something to be much simpler than you thought or feared at the beginning, then it is probably true. But if it is simple and beautiful, it must be true. Quite amazing to hear from a physicist. If you look at today's cell biology and compare it with the development of molecular biology had simplicity and beauty. Cellular biology, and especially biomedical research today, has no simplicity and very little beauty. Mainly, it is dominated by technologies rather than knowledge. Technologies as the tools for collecting data. We are choking in the excess of non-understood data, details. We learn in details something, how something is happening, but we don't know what is happening and why it's happening. And that can't assure a faster or more productive development of the biomedical research. One of the reasons is that there's little diversity in biomedical research in general. It's based on collecting data, measuring what changes, goes up or goes down with aging, compare young and old, or in disease, compare healthy and sick person. And as soon as a difference is seen, and there are countless differences to be seen, then randomly an observation of a biomarker, something that changes, it calls for intervention to target drugs to biomarkers. And what are the biomarkers? They are, in huge majority of the cases, almost always, they are so numerous because there are numerous consequences of the initial cause of disease or initial cause of aging. And it grows because the processes are so interconnected. Well, that brings us to the situation where right now, maybe everything will be clear or much clearer in 10 years, we'll see, that we are basically choking in non-understood amount of data, and therefore we are longing for artificial intelligence because apparently our natural intelligence can't deal with that amount of data without the lead of concepts and principles. The concepts cannot be complex or complicated. No concept is complicated. It's not called concept then. No equations are simpler than Newton's and Einstein's, but they give the lead, and you can derive the knowledge of complexity and derive complex equations from these principal ones. We are far in biology, in cell biology and in biomedical research from such a situation that the physics found itself. Right now, we talk and read and mention maybe 100 or 1,000 times more frequently technology, which is a tool, than knowledge, which is the purpose of the use of the tool. And that's a phase. It will pass. It will become too boring, and something else will come to make us understand this huge amount of data. Now about aging and disease. In order to get the feeling of a real discovery of what Maynard Smith called major transition, we need to be surprised. If what we found is not a surprise to the scientists who found it, then that discovery is probably not as great discovery. A great discovery is so new, unexpected, that it is a surprise. And how would we support, catalyze the number of surprises? How can we make a business plan for the production of surprises? Who would invest in surprise? Well, very few people. I try to do it here, and it is naive it gets to be hard, but again, I hope that even this experiment will be, this institute will one day be a surprise. One of the ways to describe, to summarize today's knowledge, growing knowledge with brilliant papers on aging and diseases, is that diseases and aging are very complicated, complex. There's no way to find simplicity in something so complex as disease and aging. But this is not true, I will argue, that the cause of disease and aging, I would argue, is simple, is measurable, it can be monitored, it can be acted upon. What appears complicated are the consequences of disease, the consequences of aging. And the complexity, apparent complexity, is not diagnostic of the complexity of the cause of disease or aging, but it is the diagnostic of the complexity of the consequences. In a way, the complexity, apparent complexity, is diagnostic of the complexity of the healthy organism. When we study diseases and aging, then that complexity which appears is diagnostic of the complexity of healthy organism and healthy system in general, a complex system. A simple damage occurring to a simple system will have a simple consequence, whereas the same simple damage to a complex system will have complex consequences, and we'll call it that it is extremely complicated and complex. So this is banal in a way. What people expect from us scientists in this area is that we solve their problems, that we get to such kind of knowledge that when used, it will be efficient in relieving them from suffering, from disease, to prevent disease, or if that's the best, or if not prevented, that it can be cured, to slow down aging. That's what people expect from us. And for that, what we need is to learn about the origin, about the root cause of disease, and not of its banal consequences. And then only target and act upon the initial root cause that will cause the avalanche of more and more complex consequences, because the system is so complex. Our organism is so complex, and its functionality is so interconnected. Well, I wish to propose here that upon at least 15 years of research in this, perhaps the most beautiful institute in Europe, MEDILS, Mediterranean Institute for the Study of Life, that the cause of aging and of all age-related diseases in their diversity and complexity, that root cause is simple. And second, that that root cause is common to all age-related diseases, and again can be measured, monitored, and acting upon. If there is a slight chance that I'm right about this, that would make a huge difference in the way we do research, and huge difference towards the success in mitigating age-related diseases and slowing down the process of aging. If there were a common cause to all age-related diseases that in human population branches into different particular diseases, and I'll come to this, then mitigating that common cause, it would be simpler to mitigate and eliminate all age-related diseases together, simultaneously, than to pick any single one of about 100 malignant and 100 degenerative diseases, than to pick any single one and cure or mitigate or prevent their disease. It would be easier to solve all problems than to solve a single one. Well, that's a challenge, whatever the probability of this to be true. Now, why would we think that there would be a possibility that there is a single cause to all diseases, and that the possibility exists to solve all of the problems, to stop, to prevent all of the disease together? Well, it starts already with epidemiology. On the so-called Gompertz curve, well, even today, the newborn children are at higher risk of dying than the children at age of 10. But at age of 10, by the way, the lowest mortality rate in human population, when you look from birth to 100 years to the end of life, and look at the whole population, the lowest mortality is at age of 10. From birth, it drops down, and it's lowest at 10, and then increases mathematically. It's a Gompertz law. Exponentially, with age, it increases with 5th power of time lived. Highly exponential catalytic way of acquiring diseases and dying. Now, when you look for particular diseases, what is the Gompertz curve for cardiovascular diseases, for colon cancer, for liver cancer, and neurodegenerative diseases, and so on? They all increase with age with the same 5th slope, very similar slopes, and they are so different. A colon cancer and a heart attack are so, or liver disease, they are so different diseases, and yet biological clock of these diseases that are age-related, that increase exponentially with age, biological clock seems to be the same. It ticks-tacks in humans 40 times slower than in mice. The simplest way to understand this is if it were the common cause. If it's a common clock, well, the simplest thing would be that it is a common cause. And I'll spend few minutes then to convince you that there are reasonable ideas today about the nature of this biological clock of life and of diseases, and that the cause is most likely the same for all these so different diseases, and that there is a physical basis that lets us understand why we don't die all at the same age from the same disease. Where comes the diversity in the human destiny that is already inborn, present at birth of healthy children, already present in form of benign mutations that will determine of which disease that baby 50, 60, 70 years later will die. And what is the nature of the latency period while one has to wait and do something in order for phenotype of this silent mutation to become a disease. I told you I'll be summarizing this simply to expose another way of thinking than this huge number of papers feeding, Tom Kirkwood says, up to 300 different theories of aging, and at least one, whether true or not, virtue of this one with which I will finish in 5 or 10 minutes is that it does not contradict any of these theories. It explains them and it integrates them into a story. I will be trying to tell a story on this occasion. Now imagine that we could hear and record the harmony of total biochemistry of human organism. Thousands of biochemical reactions, incredible complexity, and yet so well functioning most of our life. If we could have some kind of metrics of that harmony, it could resemble the harmony of music that we are hearing. Symphony orchestra that plays perfectly. And then say, well, in my organism, I have a matrix to measure the harmony of all of these reactions. And what would be the harmony? Well, the harmony is, the musicians would say, the harmony is the synchrony of vibrations, the sort of resonance. And I would simplify it even more, that the harmony is when there is enough of everything, of every function, no excesses. Excesses are very dangerous. They are, in general sense, metastases which break the complex system, and nothing useless. Enough of everything, no excesses, and nothing useless. Well, I've defined, at this point, I defined actually the efficiency, the efficacy of the complex system. So we find, if that is the harmony, then we find that the efficacy, optimization, is like the best music, that this is beautiful, that beauty is diagnostic of harmony and health. But if we could have this is emotions, if we could start a science of harmony, have a metrics, a general metrics of harmony, then I could direct this harmony meter towards my organism, and it could tell me, well, on the scale of 100, your organism is at 82, whatever. But we could also, such a general harmony meter, direct to the society and say, well, in France now, the level of harmony in the society is a given number, and it has increased or decreased in the last 10 years. So, while if you were to listen to the music of the biochemical orchestra of thousands of proteins, then we would notice two phenomena. One would be, with aging, we would notice two phenomena. One would be increase in noise. The same person followed from birth to 90 or 100 years. We would then hear increasing noise, but the music will be still distinct, the person is still alive. And then, along this noise, during the time, there will be disharmony from time to time. Something will break. There will be something bad happening into playing of this orchestra, and this is disease. That's what we could hear. Second, in average, 4 persons in 10,000 persons, there will be only increasing noise, no disharmony. So only aging, and I'll come to the chemistry of it, only aging, increasing noise, but no disease, no break in harmony. And these people are called centenarians, since they have no disease. They die one day, they extinguish, they don't have any more energy, gasoline, whatever, one doesn't know. They just get extinguished without suffering of chronic diseases that kill vast majority of us. And the reason is that the age-related diseases are the causes of 90% of mortality certainly in Europe and in the United States, and similar actually in the whole world. 90%. Now, when one doesn't get any of these diseases, you eliminate 90% of mortality, and of course, these persons live longer or much longer, and we call them centenarians. Studying centenarians may be the shortest way of learning how to mitigate all of the diseases. Now, what is the chemistry of noise, of aging? We found that it is the diffuse oxidation, corrosion, rusting of proteins. Molecules, there are thousands and thousands of species in each cell, species of protein molecules, and they're highly specific and faithful, precise in their activity. And when they rust, like other materials, but they are more sensitive to that, their precision activity is progressively ruined. And the chemistry of disharmony, of disease, is actually the same. It only appears, we have seen it, is due to oxidation or corrosion of one particular protein or of very few proteins in an excessive way. Now, this is the result of 15 years of research in this nice place. And it did not really surprise us that direct chemical damage to proteins, just as change or damage due to a mutation in the gene, which causes one amino acid of many to be replaced by another one, hits the function, because proteins carry on directly or indirectly all functions of life, including, repairing DNA, replicating DNA, reading DNA, making proteins, and synthesizing all of biomolecules. So no surprise that when such fine functions are damaged by rusting, by oxidation, that their functioning changes. Their functioning changes in the following way. It leads, we measured this in bacteria at least, it leads to malfunction. An oxidized protein can often malfunction. Another oxidized protein, depending where the oxidation is, it will lose its function. It's as if there was a deletion in the gene. In the third case, the oxidized proteins will become sticky and start forming rings or oligomers, which are hydrophobic and go into the membranes and poke holes in the membranes. They become toxic to the cell, to all these fine, important membranes in the cell, and to the outer membrane, which makes cell a cell, which with these holes of these toxic proteins now will be killed. It's a suicide by one's own bad proteins. By studying, I have to finish slowly, by studying extremely robust, death-resistant organisms, the freaks of nature, over almost 20 years now, we came to the same conclusion that in order to resist 1,000 times higher radiation dose than my cells, these bacteria or animals succeed in ignoring being irradiated with huge, excessive, lethal radiations by inventing ways to protect their proteins, not to eliminate all of the free radicals that cause the oxidation, but to protect the proteins. They are called chemical chaperones. Chaperones used to accompany young girls to protect them from potential lovers. But here, these molecules attach physically only to the proteins and have a close protection of them against free radicals oxidizing them. And this is the common method by which bacteria and some animals become death resistant. You protect your proteins, well, your functions are unchanged, perfect. Whatever else is wrong can be repaired by intact functional proteins, and there is no reason to die. So this is now the vision, to steal these molecules from death-resistant simple organisms, learn from them, maybe even try to see whether they would go directly, some should, into our own cells, and imitate them in order to become robust ourselves, in order to become centenarians. If this were the way to mitigate diseases, to prevent and cure even diseases. Then when we find a way to protect our proteins like some much simpler organisms do extremely efficiently, we would eliminate diseases, we would become all healthy, productive centenarians, and that this is not by itself a totally crazy, mad thought. We have a strong argument, the existence of centenarians, people who live, don't get any serious chronic disease, and by the virtue of that, live 100 or 105 or 110 years, live to be centenarians and supercentenarians. So my modest project is, let us be all centenarians and be healthy and productive to 100 or beyond. Thank you very much for your attention.